Hello, my name is Tom Sheehan. I'm a graduate student and recently elected student counselor to the Coleoptera Society. Today I'm going to talk a bit about my PhD research, which I titled Beetle Diversity in the Imperiled Longleaf Pine Ecosystem. I'm a PhD student at the University of Georgia, which is located in Athens, Georgia. And I conduct my fieldwork at the Jones Center at Itchway, a nonprofit ecological research center in southwest Georgia. I am co-advised by Dr. Joe McHugh in the entomology department at UGA and Dr. Keir Klepsig from the Jones Center at Itchway. Dr. Mike Ulishin from the Forest Service and Dr. Chris Brayman from the entomology department at UGA serve on my committee. I wanted to take some time to talk about where I do my research, which again is the Jones Center at Itchway. It exists on about 30,000 acres, which is primarily dominated by longleaf pine forest. Longleaf is very important for many reasons, but primarily its immense diversity. It is one of the most species-rich plant communities in any temperate region. Ground cover diversity here can be as high as 40 species in a square meter, and 140 species in 1,000 square meters. Endangered species live in this ecosystem too, such as red-cockaded woodpecker and indigo snake. Much of this diversity is due to frequent fire that keeps the canopy open. Arthropods here are less known, but still tremendously diverse. A few years ago, Kieran and I reviewed over 500 peer-reviewed and gray articles. And then part of that, we tabulated all of the species reported in longleaf. In total, we compiled over 3,000 species, but this is certainly an undercount, as that doesn't include arthropods never reported in the literature, arthropods never determined to species, and arthropods never actually collected. And all of these gaps in our knowledge are concerning because this ecosystem is in peril. It used to cover 37 million hectares across the southeastern United States, about the size of Montana. But it has since been reduced to just 1 million hectares, less than the size of New Jersey. This is due to many reasons, like habitat alteration, conversion to other pine species, storm damage, but also fire suppression. This image here to the right from 2003 is when prescribed fire was on the landscape, but then fire was experimentally suppressed. By 2009, the hardwoods begin to encroach, and by 2014, the canopy begins to close. This reduces the ground cover diversity and other diversity in the system. Beetles are diverse and do important things. I'm preaching to the choir here. I don't need to get too much into why beetles are important. But I do want to go over why beetles deserve attention in this system. Surprise, surprise, they're incredibly diverse. In our 2022 review, we found 84 families in 643 species of beetles. But again, this is certainly an undercount of what actually exists there. Some families only have a single representative documented in longleaf. These include somewhat surprising families like Dermestidae, Eucnemonidae, and Trochidae. These families certainly have more representatives out there, they just haven't been reported yet. So we care about beetle diversity. We want to know what species are out there. But we can also use beetles to answer ecological questions along the way. Such as, how do burn regimes influence insect communities? Pyrodiversity is the variation in fire characteristics over time and space. If an entire property burns all at once, that has low pyrodiversity. But frequent, low-intensity prescribed fires that carry across the landscape in a heterogeneous manner have high pyrodiversity. Recent studies have demonstrated a link between pyrodiversity and pollinator diversity, as well as bird species diversity. But this has not yet been examined in beetle communities. Until now. To address this gap, we set out traps in areas with several years of known burn histories. We used these white bucket flight intercept traps to target cerambycids. We sampled in longleaf of the coastal plain, as well as montane longleaf pine in northern Alabama. We'll also be looking at bees and other pollinators, but I won't bore this crowd with that. So we collected thousands and thousands of beetles. I'm currently going through the laborious tasks of processing, mounting, and determining to species all the cerambycids. But here are some that we collected in high numbers or are just particularly beautiful. So we have Neoclitus acuminatus, Molorchus bimaculatus, so there's thousands of that species. Plinthocilium suavalens is just absolutely beautiful. And Ancylocera bicolor, really, really gorgeous. And Euderces piscipes. I also caught so many Conatellus, which is presumably attracted to the white bucket of the trap. So stay tuned for more results of this project in the future. Moving on to another question. What roles do fire and termites have on wood decomposition in saprophytic beetle communities? Termites create galleries in wood that perhaps increase the surface area and aeration for the fire and might create an interaction effect on wood decomposition. They also might compete with saprophytic beetles, or they might open up additional niches. So these interactions are pretty unclear. And so this is actually building off a study from a few years ago that Mike Ulishin and I did. So to examine this potential interaction, we cut down longleaf pines into logs, exposed half of them to termites, the other half we excluded from termites, and then we waited 2.5 years, and then we exposed half of each of those groups to a fire, and the other half not. We found no significant interaction, but this was just one burn. Also, one log with termites present, it completely combusted. We did find significant mass loss due to the termites, but the burn, these are pretty low intensity prescribed burns. So to build on this study, we will add a few more levels of complexity. First of all, we will compare longleaf and live oak, so two different tree species. We will also have additional burns, and then we will also examine beetle communities as a response variable. So currently, right now, the logs are out there sitting in the woods. We're letting them do their thing. Some of them have been exposed to one burn. And then in 2026, with that final burn, we'll then throw the logs in rearing chambers to see what beetles emerge, and then eventually take the mass of the logs. Okay, so remember when I said this system has 84 families reported in the literature as of 2022? Well, that's actually already out of date. A project as part of Royce Dingley's masters down at HOA yielded an adarid from a malaise trap up in the canopy of longleaf. So a lot of sampling up to this point has been, ground-based traps, but there's a lot up there in the canopy yet to be found. So this family had not previously been documented in the longleaf pine ecosystem until this project. So this leads me to my final chapter question, which is what beetles are in the canopy, and how do these communities differ between tree species? And I would like to do this with canopy fogging. So to sample insects with canopy fogging, an entomologist will run a fogger with a relatively tame insecticide like pyrethrin to knock down insects into some sort of collecting device, such as a funnel. This has not been done in longleaf. There was one study focusing on red cockaded woodpecker prey, focused on the boles of trees, but not on the canopy. I feel very fortunate to have received the Coleoptera Society Graduate Student Research Enhancement Award to help fund the supplies for this project. For this study, I want to compare longleaf pine, which you've heard a lot about already, and post oak, which is a xeric oak typical of the system. The plan is to sample each tree species 5 times, then sample again 3 weeks later to examine recolonization. This recolonization aspect has not been studied much or at all, but we faced many logistical hurdles to address regarding getting access to the canopy. Field stilts, for example, were considered too dangerous, as was a human pyramid. Thankfully, we had access to a hydraulic lift that can take us 40 feet above the ground. Here is a time lapse of me fogging the canopy, wearing appropriate PPE, of course. After waiting 2 hours for the beetle rain to stop, we sprayed them all off the tarps into a bucket. We tried a super soaker at first, but sadly that wasn't as effective. I also took LiDAR scans of every tree canopy that we sampled. I'm working on processing the beetle samples and trying to figure out the logistics to sample again in 2025. I am incredibly grateful for these institutions and funding agencies, including all of the wonderful coleopterists of the Coleopterist Society, and numerous individuals who have contributed so much towards this research. Hopefully I have piqued your interest some. Here's my Blue Sky handle if you'd like to add me there, and thank you so much for your time.